A ver.
Muchísimas gracias, Edgardo. Sean todos muy bienvenidos. La Secretaría Ejecutiva del Consejo de Ministros de Salud de Centroamérica y República Dominicana, SE-COMISCA, la Red de Laboratorios Nacionales de Referencia de Salud Pública de Centroamérica y República Dominicana, REDLAB, el Instituto Costarricense de Investigación, Enseñanza en Nutrición y Salud, INCIENSE, y el Proyecto de Salud Global de la SE-COMISCA están invitándoles muy cordial a estas capacitaciones virtuales de laboratorio que son sobre enfermedades infecciosas. Esta es la sesión número 12 que se corresponde a la vigilancia de la resistencia antimicrobiana en el marco de una sola salud. Para esta sesión contamos con la participación de dos expertos, la doctora Sofia Ribeiro, que presentará el tema de la experiencia de Dinamarca en el desarrollo de la vigilancia de la resistencia a los antimicrobianos en el marco de una sola salud. De igual manera nos estará acompañando el doctor René Hendriksen, quien presentará la vigilancia genómica de la resistencia a los antimicrobianos bajo siempre el enfoque de una sola salud. Este es un espacio pues, que nos permite y está orientado a compartir experiencias y acciones que se implementan para fortalecer tanto la respuesta desde los laboratorios y el sistema de salud. El objetivo de estas actividades es fortalecer los conocimientos y capacidades de los profesionales de la salud, laboratorios en vigilancia de la resistencia a los antimicrobianos, todo esto bajo un enfoque de una sola salud, mediante el intercambio de experiencias, estrategias y avances en vigilancia genómica, así como también los sistemas integrados de monitoreos implementados a nivel internacional. Para esta actividad nos acompañan también el equipo de Inciensa, quien estará moderando la actividad. Nos gustaría, si se encuentra presente en este momento, la presidenta pro tempore y la licenciada Yvonne Inver nos pueda compartir algunas palabras de bienvenida. Adelante, licenciada Inver. Creo que tiene problemas de conexión. Lo vamos a dejar en el transcurso de la actividad que ella pueda presentarse.
Y
O está aquí,
buenos
ya.
días,
Adelante, adelante. Muchas gracias.
muy buenos días a todos y todas. Es para República Dominicana esta oportunidad de cada día aprender más y más en estos momentos. Sean todos bienvenidos y bienvenidas a este hermoso webinar. Muchísimas gracias a todos.
Muchísimas gracias, licenciada Ivonne. A continuación, y para darle seguimiento al desarrollo de nuestra agenda, dejo con Anto, quien presentará a nuestros expertos y estará a cargo de moderar la actividad. Adelante, Anto. Bienvenido.
Muchísimas gracias, muchísimas gracias. Buenos días a todos y buenas tardes a nuestros invitados. Muchísimas gracias desde ya a Sofía y a René por aceptar esta invitación. Voy a tratar de compartir. Yo no hice pruebas de compartir. Vamos a ver cómo nos funciona. Esperemos todo salga bien. ¿Se ve bien la presentación?
¿Todo bien, Anto?
Es muy pequeño, es para poderlos presentar a ellos. Como ya se mencionó, estamos acá reunidos para pues escuchar a nuestros invitados sobre la vigilancia, la resistencia a antimicrobianos en el marco de una salud, un tema que es un reto para todos los países que están presentes, la implementación de esta vigilancia, y no solo en la parte técnica, sino en un área muy importante que es la parte humana, ¿verdad? La relación entre diferentes profesionales e instituciones. Y para eso tenemos dos invitados de lujo que aceptaron pues venir a compartir sus experiencias. Así que los invitamos a que se inspiren a detectar capacidades que ya tenemos implementadas y además tomar ideas de capacidades que podemos implementar aterrizándolas a nuestra situación de cada uno de nuestros países. La primer presentadora, como fue mencionado, es la doctora Ana Sofía Ribeiro Duarte. Ella es la doctora Duarte, tiene formación en veterinaria y cuenta con más de 15 años de experiencia en el estudio de la epidemiología de los patógenos transmitidos por los alimentos y la resistencia a los antimicrobianos en la cadena alimentaria. La doctora Duarte es investigadora en el Instituto Nacional de la Universidad Técnica de Dinamarca, DTU, donde es parte del grupo de investigación sobre patógenos de origen alimentario y epidemiología. Uno de sus principales intereses de investigación es evaluar la transmisión de la RAM entre animales, seres humanos y el medio ambiente. Entre sus posibles responsabilidades se encuentran lo que es la coordinación, elaboración, edición del informe anual de Dinamarca, en lo que es el seguimiento de uso antimicrobianos y la resistencia antimicrobiana del programa danés para la vigilancia de este tema en el concepto de una salud. También participa en la elaboración del informe resumido europeo sobre el seguimiento armonizado de la RAM en lo que es la Unión Europea, publicado anualmente por lo que son las autoridades europeas y la seguridad alimentaria. Y nuestro segundo invitado es el profesor René Hendriksen. El profesor Hendriksen es un destacado investigador y académico de la Universidad de TU también, donde ocupa el cargo de director del Grupo de Investigación para el Desarrollo de Capacidades Globales del Instituto Nacional de Alimentación. Su carrera se ha caracterizado por importantes contribuciones en lo que es el ámbito de la RAM y los patógenos transmitidos por los alimentos. Ha desempeñado un papel fundamental en el establecimiento de lo que son los centros de referencia internacionales de la DTU en todo lo que es el tema de la resistencia a antimicrobianos. El profesor Hendriksen es director de los centros de referencia para la ramen y genómica en cuanto a lo que es centro colaborador de la Organización Mundial de la Salud, centro de referencia de la FAO y el laboratorio de referencia de la Unión Europea en materia de piensos, alimentos, salud animal y salud pública, a los que también presta servicios de asesoramiento junto con el EFSA, el ECDC y la OIA. Y la Administración Veterinaria y Alimentaria de Dinamarca en las áreas de vigilancia, una salud de la RAM y las enfermedades infecciosas. La labor del profesor se extiende a nivel mundial. Vamos a ver en sus presentaciones cómo va más allá de Dinamarca y Europa para ambos y sus esfuerzos por desarrollar la capacidad de los laboratorios han tenido un profundo impacto en las iniciativas de salud global y principalmente en lo que son los países de ingresos bajos y medios. Las presentaciones que ya ustedes vieron tanto en los temas que recibieron para inscribirse en el webinario y que ya fueron mencionados por Esmeralda. Así que vamos a iniciar con nuestros invitados. Aprovechen y hagan preguntas. Las preguntas, muy importante. Vamos a hacer las dos presentaciones seguidas. Primero la doctora Duarte, luego el doctor Hendriksen y al final vamos a dejar el espacio de preguntas donde estarán apoyando también Tabata Lizondo de por acá de la Inciensa y Esmeralda desde Comisca. Muchísimas gracias, doctora Duarte. The floor is yours.
You. I hope it will work now. Sharing my presentation. One mode. Can you see it in presentation mode?
Yes.
Yes, perfect. Just like to remove this here. Okay. Hello, everyone. I'm very glad to be attending this webinar and to have the opportunity to talk to you about the Danish model for AMR and AMU surveillance that counts 30 years of existence celebrated this year. And I would like to thank the organization for the invitation to be here. I'm not sure how much René will talk about our institute. This is a very, very brief overview about the National Food Institute that is part of the Danish Technical University. We have about 14 different research groups, and we do a little bit of everything that a university should do, which includes scientific advice, education, and innovation. I'm sitting in a research group called Foodborne Pathogens and Epidemiology, and part of our work are a lot of advisory tasks to the Danish authorities, including on the action programs for the control of Salmonella and Campylobacter in food-producing animals, ad hoc advisory on foodborne zoonotic risks, monitoring or development of monitoring methods both for food safety and for AMR. And then we are part of two large national monitoring programs. One of them is Denmark, which you will be hearing more of in this presentation. The other one is the annual zoonosis report. So it's a surveillance report on the zoonosis, occurrence of zoonosis in Denmark. I also have colleagues who sit in the Central Outbreak Management Group for Foodborne Pathogens. It's a intersectoral group that investigates foodborne outbreaks in Denmark. And we integrate also several networks, both at EU level and at international level. About me, I'm not saying more because it was a very nice introduction. The outline of this talk will be about what is One Health surveillance and integrated surveillance, and how Denmark is a model of integrated surveillance, and what are the strengths and limitations and needs in order to establish and maintain such an integrated surveillance program. Starting with One Health, the definition of One Health. One Health can be defined as a collaborative, multi-sectoral, transdisciplinary approach that recognizes that human, animal, and environmental health are interconnected. It promotes the health and well-being not of one domain, but all three, so animal, human, and environmental health, and emphasizes the need for collaboration and communication among different disciplines and sectors in order to achieve the optimal health outcomes for all the domains. Integrated surveillance has a rather overlapping definition with a slight difference. It also relies on collaboration and a cross-sectoral approach. It involves the collection and analysis of data from multiple sources, and it may, but does not necessarily include all the One Health domain. So it is possible to have an integrated surveillance program without including all the three One Health domains. Obviously, what we wish for all around the world, I dare to say, is to have both integrated and One Health surveillance, so a surveillance system that encompasses all the domains, animal, environmental, human, and also food, and that integrates data both on antimicrobial resistance and antimicrobial use or residues from each domain in a cross-sectoral approach. This is borderline utopic, utopical, how do you say? But it is possible to work towards this goal. And this is what Denmark has been doing for 30 years now. Back in 1995, the Danish Integrated Antimicrobial Monitoring and Research Program, abbreviated as DENMAP, has been established. It has produced an annual report since 1997, and it has been from the beginning an intersectoral collaborative project among the animal sector, between the animal sector and the human sector. The institutes and their affiliation have changed over time. Obviously, in 3 decades, a lot has changed, but in essence, the same sectors have been represented, and at present, the institutes that form the core or the main editorial group of the DENMAP program and report are the National Food Institute, where I'm sitting, and Statens Serum Institute, which is the Danish Public Health Institute. There are many other contributors to the program, but we coordinate the program and make sure that there's an annual report. The program covers AMR occurrence in indicator bacteria from healthy animals, in zoonotic bacteria from animals and humans, and in pathogenic bacteria both from humans and animals. And it covers the antimicrobial usage in humans and animals at 100%, so all consumption of antimicrobials in Denmark since 2002. And it celebrates 30 years, as I said before. Denmark was already back then, before the One Health concept was invented, so to say, or so widely disseminated. It was born already integrated. And this was because in the mid-90s, there were concerns in Denmark due to the widespread use of growth promoters, antimicrobials like avoparcin in animal production, and the rise in macrolide resistance in Enterococcus from human infections. So the Danish government decided back then to fund a monitoring program to monitor both resistance and consumption in both sectors, the animal sector, in food-producing animals specifically, and in the human sector. So the foundation of the program was, and it continues today to be, a program that informs and supports policy change in the matter of antimicrobial use and resistance. And the background for implementing such a program and for the government to fund such a program was that the use of AGPs, antimicrobial growth promoters, was approved at EU level by the European Commission, by a committee called SCAN. And Denmark was only allowed to ban the use of avoparcin as AGP if they could provide information that was evidence that there was a danger to human health, there was a hazard to human health to use AGPs in animal production. However, with the little data that was available, it was only originating from research projects. This direct risk to human health was difficult to prove, and more continuous and harmonized data was needed in order to establish this link. So Denmark, as I said before, has been since the start a cross-sectoral collaboration, and it continues being so. And here in this slide, you can see the participants and the stakeholders that form part of the Denmark consortium. As I said, Statens Serum Institute and DTU form the core editorial group, which you can see. I don't know if you can see my mouse moving over the presentation.
Yes.
You can. Perfect. Thank you. Yes, so this forms the editorial group. This editing group, even though we sit in different institutes, we have different backgrounds, we meet quite often and we work as a team. So we meet each other once every second week, sometimes every week towards the end of the annual cycle. So we really work, even though we work with our own data separately, we discuss the results and try to do integrated discussion of the results and interpretation of the general picture of what is going on in Denmark in terms of AMU and AMR. This is only possible because we meet regularly and we work as part of one team. We also interact with other units within our institutes. For example, I interact with René's group when I need their expertise. There are a lot of experts on microbiology and genomics sitting in René's group, also other groups at DTU. And I'm not a microbiologist, I'm not a molecular biologist, so whenever I need this expertise input for Denmark, I go to other colleagues with the right expertise at DTU. And my colleagues at SSC, they do the same if they need some expert knowledge on or expert input on one or the other pathogen, for example. Then we interact with our stakeholders at the Danish authorities, so the ones who actually own the data, and the ones who are interested in the output of Denmark to do new policies, for example, for the control of antimicrobial usage in the different sectors. And we interact with the authorities on biannually, perhaps a bit more often. Usually it's around the time we receive the data from the authorities, when we have preliminary results, and then when we are about to publish the report to make sure that they're informed about what is going to be published. We also have general meetings with contributing authors, both in spring when we have the first results available. So we sit all together and discuss the results across both sectors. So the colleagues from SSI, the contributing authors who know more about human pathogens, they also hear about the results of what is going on in the animal sector, and we have an active discussion at this spring meeting. And the same happens then at the annual seminar that we hold every November when we publish the report. And there we invite not only contributing authors, but all sorts of stakeholders, mostly national. But for a couple of years now, we have also hold a hybrid meeting where international colleagues are also welcome to attend, because some of the presentations we try to keep in English. We also have a steering committee with whom we meet twice a year, and this includes colleagues, 2 colleagues from DTU, 2 colleagues from SSI with expertise on AMR and a long knowledge of what the Denmark program is and what it entails. So whenever we need to discuss the direction or of the program or any challenges we encounter, we meet with the steering committee as well and receive their input. So it's a very active collaboration between the 2 sectors. The 3 main aims of the Denmark program have been the same since the start, and they are to monitor antimicrobial use and resistance in both the human population and the animal populations. When I say animal populations, I refer mainly to food-producing animals, to study the associations between usage and resistance within the sectors and across sectors, and to identify routes of AMI transmission. So I think, I hope by now you're convinced that we have an integrated surveillance program in Denmark, but how One Healthness is this program? Because as I mentioned in the beginning, One Health and integrated surveillance are not necessarily one-to-one or the same thing. A couple of years ago in 2021, we had a visiting researcher who performed an evaluation of the One Healthness of the Denmark program using this so-called One Health EpiCap tool, which scores the surveillance program on different points, so to say, different 3 dimensions and different points within each dimension. And the score 4 is the highest a program can obtain. And as you can see in this plot, Denmark scored next to 4 in almost all points, except for target 1.2 and target 2.3. And these are coverage, transdisciplinarity, and data analysis and interpretation. And this is the first one is due to the fact that we don't have the environmental sector or domain represented in the program. And the second point on data analysis and interpretation, it's because we are unfortunately constrained in the data that is comparable across the sectors. So the missing One Health element in Denmark is definitely the environment. We have food represented to some extent because we look into some types of resistant bacteria in meat sampled at retail. We also look at some salmonella in carcasses, for example. We definitely have the animals represented, the humans, but we have no environmental data. And some years back, there was a study or an assessment that found that there was no evidence that highlighted the importance of including the environment in the program. However, since then, a lot has happened in Denmark, and the livestock production has intensified, the farms are bigger, and the animals are also produced under different conditions, and the human hospital activity has also increased. So an EU assessment is needed, and things in the EU are also moving towards environmental monitoring, so we definitely, or most likely, will need to go there as well. But we are challenged in many ways. We don't really know where to start. We need to assess the evidence or produce new evidence to inform how exactly, where should we start, what types of samples, what type of environment is relevant to monitor. We lack the governance structure. We have a very well-established structure to monitor the human and the animal sector, but we lack a governance structure for the environment. We lack an operational structure as well. And we expect that we will have to accommodate new partners into a decade-long established collaboration between these two partners that I've mentioned previously. So there will be a lot of adaptation needed, and I'm guessing trial and error as well, before we have a truly One Health surveillance program in Denmark. But we do have an integrated surveillance program, and I would like now to give you some examples or explain how so. And the first way Denmark is an integrated program is because we report both data on antimicrobial use and antimicrobial resistance. In our Denmark report, which you will find online on our website, you always find this. In the latest reports, you'll find this diagram that explains the origin of the data that goes into Denmark. So on top, you'll have the data from the animal and food sector, and in the bottom, you have the data from the human sector. And then on the left side, you have data on antimicrobial consumption and antimicrobial resistance data on your right side. Starting with the AMU data, so antimicrobial use data from animals, these are data that are extracted every year, once a year, from a national database called VetStat, which is maintained by the Danish Veterinary and Food Administration. And the data that is extracted from this database is then taken by one of my colleagues and harmonized or summarized in different ways. One is in the total kilograms of antibiotics, but also different active compounds. And this data are used to make an overall crude comparison of how much antimicrobials are used in the veterinary sector and in the human sector. It's a very crude comparison, obviously. So we need normalized metrics, which we call the DADD and the DAPD, and specifically the DAPD, which corresponds to the proportion of an animal population that is in treatment in any given day. We use to monitor trends of the different active compounds in a specific animal population, and for some populations also within a specific age group in that population. So when you see the APD in our plots, this is normalized by both the dosage of the active compound for a specific indication and also the biomass of a specific animal population or an age group within that population. And here you see some examples of how we show those data. If we are reporting on total antimicrobial consumption, we usually show this in this pie chart, and it's always, I think, enlightening to see how, and it shows why it is important that we normalize the consumption data by biomass, because you see how the biomass is distributed on your left side. We could say it's almost half-half between cattle or young cattle, calves, and pigs. Of course, there's more, but a large share of the animal production or animal biomass in Denmark is covered by these two classes or categories, whereas more than 80% of the antimicrobial use corresponds to use in the pig sector. We report on antimicrobial class level. You see on the table in the bottom. You can see also that this is for the category weaners, so pigs, fattening pigs with less than 30 kilos of live weight. But as I was saying, we report at antimicrobial class level, so this would be aminoglycosides in the first example, but also at specific compound level. And the trends, this is different years, so the consumption in different years, and the trends can look quite different for different compounds within the same antimicrobial class. When we look at trends overall for the different age groups, for example, in the pig production, we usually group the data by antimicrobial class. Otherwise, we would have thousands of plots in the report, which already counts with 200 pages. But we can always couple the trends with then the data in the tables that show that the trends can look different when we look at different compounds. Moving on to the antimicrobial consumption in humans, the data is also originating from national databases, specifically the one on top called Sundhedsdatastyrelsen, or the data from Sundhedsdatastyrelsen, that is the data provider, who collects in a centralized way data from sales of antimicrobials in hospital pharmacies to hospital departments and wards, sales from private pharmacies to citizens, and also collects data on the National Patient Registry, so different information regarding the patients who were treated with antimicrobials. And then we have, or the colleagues at SSI also collect data from Denmark's statistic on demographics, for example, the different population in different regions of Denmark or different municipalities. The usage data is, again, on the human side, summarized using different metrics, and the metrics are different depending on whether the colleagues are summarizing antimicrobial consumption for primary healthcare or for hospital use. And you can see here some examples, and the plot also shows for primary healthcare, for example, how the trends can look different depending on how they are summarized and which metric is used. And there's always a discussion in the DenMAP seminar with medical doctors on, there's always a preference depending on the sector, their specialization, which metric they will prefer to look at. Some examples of how the antimicrobial consumption data are reported using these different metrics. Overall, the use is reported separately for primary healthcare and hospital care, and you can see that the large majority of the use in Denmark corresponds to use in primary healthcare. The colleagues also summarize the use for primary healthcare at a municipality level and also how it has changed over time. And then they summarize the occurrence or the consumption, overall consumption grouped by antimicrobial class, just as I explained also for the animal sector. And additionally, for the human sector, they group the antimicrobial compounds by the aware categories from WHO, so the access, watch, and reserve. And there are specific targets in the EU in order to, or regarding how much, what proportion of the use within a certain EU member state should correspond to an access or watch or reserve antimicrobials. Moving on to the monitoring of antimicrobial resistance, as I said in the start, there are three categories of bacterial agents monitored in Denmark. They are pathogenic agents, both on the human and the animal sectors, which primarily will reflect resistance that is caused by the use of antimicrobials in the respective reservoirs. Then we have the zoonotic bacteria that can develop resistance in the animal reservoir and may subsequently compromise treatment in humans, and indicator bacteria from healthy animals, because these are traditionally E. coli, because it is ubiquitous in animals and humans, and they can develop antimicrobial resistance when there is selective pressure, and they can transfer resistance genes to pathogenic bacteria. Some examples of how we report AMR in bacteria from animals in food. We report with this traffic light plot on the relative occurrence of different resistance profiles, which can be actually fully sensitive, so complete susceptibility to all the tested antimicrobials. It can be particular combinations of resistance, like combined resistance to ciprofloxacin and tetracycline and Campylobacter jejuni. This can vary a bit what we report depending on the bacterium of interest and what combination or multi-drug resistance profile is of interest to monitor in that bacterium. We also report the proportion of isolates that are resistant to different antimicrobial classes or compounds, like in the plot here that I'm showing. We also monitor in meat specifically, because it is mandatory to monitor at EU level. We monitor the occurrence of extended spectrum beta-lactamase producing Escherichia coli, also carbapenemase producing, even though we don't find those in Denmark, fortunately. So for that category of bacteria, we monitor both the phenotype, but also we sequence them and we monitor what is the genotype behind the ESBL or MC phenotype. Moving on to the resistance data from humans. Very shortly, how the data is obtained. The data reported in Denmark represents samples that are collected both in general practice, so the primary sector, and at hospitals. They can be examined either by the regional departments of clinical microbiology, or if it's a specific pathogens, the isolates can be sent to the reference laboratory, which is also at SSI. So the data, the actual results, antimicrobial susceptibility results, will be collected by the SSI colleagues either from a national database called the Danish Microbiology Database, MIBA, or from the specific reference laboratory database. Of course, a patient can contribute with several isolates in the course of 1 year, but the colleagues have the criterion that they will only report the results from the first isolate collected from one patient for a given year. And these are some examples of how the AMR reporting looks for the human sector. The first plot here that I'm showing is not really related to AMR. It's just a relative distribution of what agents have caused the reported invasive bloodstream infections in humans in Denmark in a given year or over time. And then for each of these agents, the colleagues will look into the trends for resistance to different compounds. They also keep an eye on more worrisome resistance types like CPE or CPO organisms. And you can see that we have this very worrisome trend of an increase in the reported cases of carbapenemase producing Enterobacterialis in Danish patients. So whenever there's a worrisome trend, then the colleagues also go and look at different metadata behind these cases, depending on the data available. This is an example where they looked at the origin of such isolates and whether they were from patients who had been traveling. In some cases, they were patients who had been transferred from Ukraine, who was while already after the start of the war, they've been transferred to Danish hospitals. So they try to investigate what can be behind such trends. And they also report for some of the pathogens the genomic or the genotype that is behind the resistance profile. And sometimes this shows that the genes that are behind the phenotype can actually change over time. So it provides more information than just reporting the phenotype. When reporting, we report, as you just saw in all of these examples, antimicrobial consumption and resistance separately, but we also, whenever possible and relevant, report in an integrated manner AMU and AMR. These are two examples where we thought it was relevant to show how the occurrence of resistance to a certain compound was following a similar trend to the increase in use of a related compound. So the first example was in Danish pigs and pork, where we know that the use in apomycin has increased in wieners, and we saw an increase in resistance to gentamicin in isolates from Danish pork. And a similar example here on the right, where there's been an increasing trend in the use of fluorphenicol in calves, in calf production, and we also saw an increase in chloramphenicol resistance in indicated coli isolates from calves. This is a similar example, but just to show that this data, from this previous example, they are all sitting, so to say, at DTU. So it is our responsibility. So I just need to talk to my colleague who reports the antimicrobial use data in order to produce such plots. But we also do it across the two different institutes when it comes to pathogenic bacteria from animals, because even though it's the animal sector, the colleagues who are reporting resistance trends in clinical isolates from animals, they are actually located or affiliated at a consortium called DKVET, but they are sitting at SSI. So we also communicate across institutes whenever the colleagues see a worrisome trend in their clinical isolates from animals. Then they ask us, did you see a trend in use that could justify this? And then we discuss the data in the report in an integrated manner. And the colleagues on the human sector do exactly the same, either to highlight some worrisome trend, just like as I just explained, but also if trends are going in the right way, in the way that we hope for, that is also worth showing. DenMap is also integrated in a way that we do within the sector reporting, but also across the sectors. And you just saw an example of how we communicate across the institutes, but we also communicate between the animal sector and the human sector. We have all of these chapters in the DenMap report, and the ones on the right that are circled here on the right, they are to report data within one sector, whereas these two chapters on the left, on the right, on my right, so chapter 6, resistance in zoonotic bacteria, and chapter 3 on One Health EMA, here we do intersectoral reporting, as well as in our little brother of the large report, which we call the summary report, which has been produced since 2021, I think, or 2020. So some examples are in chapter 6, which is the chapter on AMR in zoonotic bacteria. We report resistance trends or resistance occurrence in both Campylobacter and Salmonella in both humans and food-producing animals. We prepared this chapter in close collaboration with our SSI colleagues, where we put the data in the same tables and the same plots, and we also discuss it in an integrated manner in the text. And here we report separately for different Salmonella serotypes and different Campylobacter species, because they can present very different AMR profiles. Also in this chapter, we focus on trends of occurrence of resistance to specific antimicrobials that are relevant in the Danish context, in European context as well, for human treatment of infections with these pathogens. And this is to warn for concerning trends, which we definitely do not wish to occur in the animal reservoir, because they may reach the human the humans at some point, and also to identify signals for possible zoonotic transmission of resistant Campylobacter and Salmonella. In Chapter 3, which we call the One Health AMR, it has become a ESBL E. coli chapter, and this is mostly because when we, in 2021, I think it was, we sat down, the TU and SSI, and we looked at the collection of data that we usually have available to report in Denmark, and we concluded that apart from Campylobacter and Salmonella, the only bacterium where we had comparable data to some extent was whole genome sequencing data from ESBL producing E. coli isolates, or MC producing. So we started back then to investigate the overlap between the combination of MLST type and the occurrence of different ESBL genes or MC mutations in these E. coli isolates that we had both from food-producing animals, retail meat, and humans with bloodstream infections. Now, of course, a human with a very serious bloodstream infection is perhaps very far in terms of occurrence of E. coli. A lot can happen. So it is a very crude manner of comparing or assessing whether there is a zoonotic transmission of this bacterium, but unfortunately we don't have genomic data of healthy humans or humans with other types of infections, such as urinary tract infections, for example. So this is what is possible given the data that we have available, and we have produced this plot, which shows that, at least for this data set, we cannot find a clear evidence of zoonotic transmission. We also report in our summary report, which is a smaller version of the full report, we report the antimicrobial use across both sectors, both with the total use of antimicrobials, so tons of active compound, but also divided by different animal species, and normalized by biomass, so DAPDs for animals and DIDs for humans. And the last example of how Denmark is integrated is related to the fact that it integrates data from both passive surveillance systems and active surveillance systems. So I'm just going to refer to the data that refers to animals in this case, or AMR from animals. And here DTU reports on data from an active surveillance program in healthy animals at slaughter and retail meat, whereas the colleagues from the DK Vet Consortium, who are affiliated at SSI, report data from a passive surveillance program from diseased animals. The active surveillance program collects different bacteria from healthy animals and food, and it fulfills, or most data comes from fulfilling the requirements of the EU legislation on the harmonized monitoring of AMR. And this legislation establishes that in odd years, or all member states should monitor AMR and bacteria from pigs and calves, and at slaughter, and the pig meat and calf meat at retail, and in even years it's about poultry, so broilers, laying hens, and fattening turkeys. And the bacteria are the same. It's just the animal species that rotate. And this is what is mandatory, but member states are also welcome to report on all 4 animal species or 5 animal categories every year. So this legislation also describes in detail how the EU harmonized monitoring should look like, where the samples should be collected, what animal species, what are the antimicrobial susceptibility testing methods. I guess René will talk more about that. In case the isolates are sequenced, also what kind of tools and sequencing technologies should be used, what kind of databases. So everything here is harmonized. There's a randomized sampling plan defined that all member states should be following. There's a minimum number of isolates that should be reported. and the sampling should be representative both throughout the year and also geographically within each member state. There's an extensive information exchange at EU level from this monitoring program. So we do report this nationally in Denmark, but we report this data to EFSA, or the European Food Safety Authority, every year. I am data reporter for Denmark, so now by the end of this week, I have to have all the Danish data for 2025 reported to EFSA. So each member state will have a data provider, reporting officer who's responsible for different things, data reporting, then reviewing the data that has been reported and approve it. And the approved data from all member states will integrate the EFSA ECDC annual report. And there's a strict calendar with legal deadlines that all member states that you see here, this is the calendar for the current year, that all member states need to follow. And then every year in November, there's an annual network meeting with all the reporting officers, the data providers from every member state can attend physically or remotely, but to be informed about the latest results, what will be included in the report, and come up with any relevant input. So in this active surveillance program, everything is planned and harmonized. The samples are taken at retail, at the slaughterhouse, and at border control posts. At the retail, samples are taken by the food inspection units. The samples are randomly selected, so it could be, and this is to cover both domestically produced meat but also imported meat. And a lot of information is collected on the origin of the meat, including where the animal was born, raised, slaughtered, cut, and packed. The slaughterhouse samples are taken by meat inspection units. Only one sample from each per slaughter batch should be collected in a given date. And the sampling is as randomized as possible based on the slaughter throughput of each of the national slaughterhouses. The border control posts are sampled to have a representative sample of what is imported from countries outside of the EU. We don't have that much meat coming from outside of the EU or that hasn't gone through an EU country before it arrives to Denmark. So we usually don't report or don't have that many samples from border control posts. That was an active surveillance of AMR in animals. Then we have the passive surveillance of AMR in veterinary pathogens. And this, as I said, is performed by the SSI colleagues. These isolates are collected in a passive manner where we don't have an insight on how representative the sampling is or whether there's any sort of bias. It's collected by either the private industry or sent to our SSI colleagues for analysis, either from the private industry labs or from vet practices. And then once a year, these two cases, depending on whether it's a pig reporting year or a cattle reporting year, they will send the isolates and the antimicrobial susceptibility data and eventually allow the colleagues to do whole genome sequencing on the isolates as well. This is a list, we don't need to read it, but there's an extensive list of pathogens that are monitored both in pigs, mostly with diarrheal disease, and cattle with mastitis. More sporadically, there's also reporting of pathogenic bacteria and their AMR profiles from dogs and cats, but because the data collected from this animal species is so scarce, usually we have to wait 2 to 3 years in order to have a large enough number of isolates to report in Denmark. Also for this passive monitoring of AMR and animal pathogens, there's a lot of information exchange and attempts of harmonizing the way this monitoring is done at EU level. It's not as established and it's not legislated as it is for the active surveillance in healthy animals at slaughter or in meat, but there's a project called EARS-VET that is funded by EU that has been for many years and continues to try to fill the gaps in this passive AMR surveillance in bacterial pathogens, and in order to inform in a more tailored way antimicrobial stewardship in veterinary medicine within the EU. And they're also working hard to generate minimum inhibitory concentration distributions to inform the setting of ECOFs or CBPs for interpretation of results in the future, hopefully mandatory monitoring system. To wrap up, I hope I'm not very much over time. Strengths and limitations of such an integrated surveillance. I would say that the first an absolutely necessary requirement in order to have an integrated surveillance system is a good collaboration and trust and respect between partners that sit in different institutes, have different expertise, work for different sectors and inform different authorities. They need to work as a team. And I think we can be proud in Denmark that this is the case. Of course, it requires comparable data, and you saw the example that even in Denmark, where there's an integrated program that counts already with 3 decades, we are very limited in the data that is comparable across the sectors. And then we need, of course, funding and time in order to sit people around the table and actually have the opportunity to compare results across sectors. The advantages are that it allows the coordinated adjustments and responses across the sectors, and it gives a comprehensive state of the nation in terms of antimicrobial use and resistance. But any action will not be a silver bullet approach. It will need a continuous assessment and update and optimization. And just to show you how this applies to Denmark. The Denmark program has over time, and this goes only until 2020 already, we are already in 2026, but it has given here a comprehensive state of the nation, and it has informed or supported the implementation of all of these measures that you can see highlighted or plotted here, that include the centralized or the establishment of centralized databases or implementation of either mandatory or voluntary measures in different sectors to reduce the antimicrobial use, the release of specialized treatment guidelines for different sectors or different animal species, or the implementation of the targeted monitoring of specific resistance like CPO in humans. I would say, to summarize everything that has happened in Denmark in terms of reducing AMU and controlling AMR, the first 20 years have been mostly focused on reducing the use, mostly in the animal sector, and developing treatment guidelines, whereas the last 10 years have been mostly focused on developing national guidelines and a One Health approach and specific goals and action plans that are aligned with the EU and the global plans as well, in order to bring further down the use and the resistance in Denmark. And then it has supported and also shown how the coordinated adjustments these examples are just intersectoral, but still that adjustments in reducing the use have led to an resulting reduction in the resistance as it was expected. And it is a continuous journey, so it's a program that hopefully will count with many more decades, because what we've experienced, and now just looking at the example of antimicrobial use in animals, is that whenever there's a worrisome trend, then Denmark has shown that this was the case. There was some corrective measure or control measure implemented, which brought the trend down or reversed it. But then over time, there's of course some adaptation of a given sector that maybe changes the use to a different compound, or it grows, it becomes more intense, or the animal production, for example, intensifies. So over time, then we experience another worrisome trend, some control action is put in place, it goes down a bit, and we go like this, like a little bit roller coaster. But not to be too gloomy about it. If we look over time, over this 30 years of the Denmark program, the trend has been a positive one, meaning that Denmark has managed to reduce the use of antimicrobials in animals. And this is what I had for you for now. So thank you very much for your attention. You're welcome to contact me if you have any questions, and feel free to visit our website where you'll find all the reports available in PDF. Thank you very much.
Muchísimas gracias, doctora Duarte, por la excelente presentación. De nuevo uso la palabra de inspirarse, muy inspirador y poder ver hacia adentro qué tenemos y qué también podríamos de alguna forma tomar y aplicar o aterrizar a nuestros países. Ahora, perdón, vamos a pasar a la segunda presentación del doctor René Hendriksen. The floor is yours, doctor.
Thank you, and I'll try to share my slides.
Antonieta, disculpa la interrupción. Mientras continuamos con la siguiente ponencia del doctor René, podríamos esperar un minuto de receso para que el intérprete pueda seguir con el servicio, por favor.
Perfecto. Vamos dando un minuto y si gusta, intentamos ir proyectando o...
Sí, por favor.
Doctor,
My
if you want, you
presentation
can try
in
to
presentation mode.
Yep. You can try to put in the presentation mode.
It should be in presentation mode. No?
No yet.
Okay, let me see. What if I do this? Is this?
No. We will
Okay.
wait just 1 minute. We have to adjust something.
Okay.
Esmeralda, ¿nos guías?
Sí, yo les indico en el momento que ya el intérprete esté nuevamente disponible. Mientras viene, no sé si tienes algún comentario, Anto, que puedas hacer o...
Bueno, creo que los hice como al final de la presentación de la doctora Duarte. Lo que no me gustaría es ahora, ¿verdad? Tener como los dos idiomas, pero sí, bueno, espero que no hemos visto preguntas por acá en el chat, me parece. Ojalá y se animen para ir preguntando y de nuevo, sí, me parece que no habían por ahora, preguntas.
Listos, estamos listos,
Listo,
Anto.
perfecto. Eileen, ya casi
Anto.
volvemos al final y empezamos con vos. Disculpa, ya que levantaste la mano y ya casi volvemos y te anotamos para empezar por tu pregunta o comentario. Gracias.
Adelante.
Dr. Hendriksen, you can begin with your presentation. Thank you.
Thank you. So first of all, I want to thank the organizing committee for inviting me to give this talk about one of my favorite topics. But I also want personally to thank Hendriksen for inviting me and giving this opportunity. So Henrietta also gave me some points to talk about. So I'll start a bit with our introduction, why we need genomic surveillance, what are the benefits. Then I will touch shortly about our first experience with genomics more than 15 years ago, more or less. And then talk a bit about how to set up whole genome sequencing surveillance in low and middle income countries with some data from our UK aid project called SE-COMISCA, where I'll end with talking a bit about barriers and in specific about sequence cost. Then I'll talk briefly about how we have set up the EU monitoring. Now we heard Sofia talking about the surveillance both in Denmark, but actually it also reflects how it's set up in EU, but I'll give a bit more details on that. And then also, how do we actually train bioinformaticians and prepare everybody for genomic surveillance? And then latest, how we circumvent some of the barriers with understanding genomics. So that's sort of what I want to touch upon. But why not just start with Pasteur and Fleming? So basically, we have for decades used what they discovered centuries ago, and basically funded the modern microbiology that would allow us to understand the causes and how to prevent bacterial diseases. So, of course, this started with Fleming discovering penicillin in 1928, but that really revolutionized how we treat bacterial infections up to today, basically. But today, a lot of our diagnostics is still actually based on those discoveries and concepts and methodologies with conventional or phenotypic microbiology dating these decades or centuries back. So, as you all know, we are in a crisis situation with the emergence of antibiotic resistance that threatens the very core of modern medicine. This is a quote from the former director of WHO that really put AMR on the agenda. And of course, the emergence of antibiotic resistance is especially something that we see in resource-limited countries that likely will be more impact of the consequences of AMR. So this is also the reason why we need, first of all, a sustainable, but also an effective and a global public health response to react to these emergencies and threats. And that was also the reason why WHO in 2015 announced during the World Health Assembly and requested basically an immediate action for harmonized and standardized surveillance on a global scale. And I think we have all been working on that. You had the example of Denmark, but I know that there are also other monitoring programs, both nationally, but there's also regionally, and also, of course, globally. Globally, we have in the public health area, GLASS. In the food and the veterinary area, we have also InFarm as good examples of global AMR surveillance programs. The problem is that if you do not have a robust suitability testing methodology, and you try to skip corners because of barriers, because of cost, lack of knowledge, then you very often end up with unreliable phenotypic suitability data. So this is just an example of another UK aid project that we were running in Southeast Asia with all of the public health laboratories and all of the veterinary labs that were officially designated as national reference laboratories. So we conducted over 5-6 years 11 proficiency tests or external quality assurance scheme tests where we sent out our 8 isolates per trial that target, of those 8 isolates, 5 were within the target organisms. And we covered all of the GLASS and FAO priority pathogens. But we also supported the trials with ATCC strains, but also interesting and maybe unique in this regard, also internal control strains. So not a ATCC strain, but a random strain that we, with the first EQA, selected as an internal control that we kept sending out in all of the 11 trials. And here, I know this is a bit of a busy slide, but here you can see two red bars in, what is that, the left side of the slide, where you can see that you have equa 3, 5, 7, and 9. And here you can see for enterococcus that in the first red one, you have a lab that started off with some doubtful, questionable results with deviation levels between 80 to 95%. And then they improved, and then suddenly, of various reasons, they provided unsatisfactory data, so below 80%. And remember, this is the same strain that are sent out in each of the trial. So why is it that suddenly it goes very bad? Of course, there's other examples. One that started off with really unsatisfactory, then they improved a bit, then they went bad again, and then they really improved. So again, there's this very complex picture of proficiency when you run trials year after year, and especially where you have an internal measurement like this control strain. And on the other side of the slide, you can see again we have the 9 trials by colors, but you can also see the individual labs. There's quite a spread among the labs in terms of proficiency. So again, I would argue that maybe up to 10% in an average level of suitability tests are not reliable, which is some accept it. I don't. I think 10% is way too much. In EU, we have a threshold of 5% of deviation, which we find is acceptable. But as you can see, we actually have labs that provide data that are really unreliable in that area. So this is why whole genome sequencing in surveillance is important, because data have shown that it's more robust. When you do concordance analysis between phenotypic and genomic data, it's always the phenotypic data that comes out being false or incorrect. So genomics is basically one method in all, or all in one technology, and it holds this immense potentially, especially for the research constrained countries where access to public health might be limited with diagnostic surveillance, etc. So it offers really a vast amount of information, as you probably also know, to the highest resolution of subtyping of pathogens. It really provides and enable the user to provide both very accurate but also rapid data for diagnostics. And it's been really instrumental in tracking and managing outbreaks, especially combined with sufficient metadata. So it really provides a much better understanding of complex epidemiology of both the foodborne pathogens, as Sofia just talked about, but also AMR. And it also provides us with the genetic markup, genes, mutations, but also flanking regions, transposomes, plasmids, and it would allow us to trace also the high-risk clones. So Sofia talked about the MLST types. So this is really important knowledge combined with AMR genes, combined with plasmids. Then we can actually track these high-risk clones. And this would allow us to respond quickly to outbreaks, but it would also be, we would be able to implement intervention control programs, but it would also allow us to assess if any of the mitigations have been a success. At the same time, it would also reduce our costs. So there have been plenty of papers showing that $1 given for surveillance will be returned with a surplus. So this is really also important. Again, it basically will provide us with a much better disease prevention strategy, and would provide us with improved treatment, it can be used for drug developments, but also in healthcare policies. So this is again something that politicians needs to know, because they are the one that are responsible for public health actions. And here they actually have a tool that actually provide both the means, but also the evidence of what they are trying to do. So going back to 2011, the US CDC, the US FDA, and DTU convened a meeting with the EU Commission to sort of establish a global microbial identifier initiative. So this was at the brink of sequencing going into diagnostics. So we were already at that time thinking about the paradigm shift in surveillance and basically moving from phenotypic surveillance into genomics. And this is probably one of the biggest revolutions since Pasteur and Fleming. So in 2011, it was forecasted that basically in 5 to 10 years from 2011, all clinical labs would have a sequence instrument in place and basically being able to generate bacterial genomes to the cost of €50 or 50 US. We would probably also have the capacity to exchange large data set across web-based systems and share these data in global repositories that would allow us to take action. So that was sort of forecasted already 15 years ago. Today, we have tried to implement genomic-based AMR surveillance. It's been a challenge in many regions of the world, really delaying the realization of its full potential. So we have, of course, seen a lot of barriers that I'll touch upon. But again, it's a bit of a pity because it really has this huge potential, as I just indicated. So our first experience goes back to basically your neighborhood. We're back in 2010. We in Denmark, in DTU, have just purchased our first MySec, brand new, coming out of the cradle. We had just started push some buttons. And then we saw in the news that there was this massive earthquake that hit Haiti the 12th of January 2010. We also heard later on that there was a lot of arguments who actually was responsible for the cholera that followed the earthquake, because again the UN immediately initiated aid relief, and among that aid relief, Nepal dispatched a battalion of soldiers to help Haiti. Unfortunately, there was an outbreak in Nepal already half a year before the earthquake in Haiti. So there was already from the beginning, with the first evidence of a cholera outbreak in Haiti, some speculations if it was the Nepalese soldiers. So we were actually able, as a WHO collaborating center, to reach out to colleagues in Nepal and have them to share the isolates from the Nepalese outbreak that was ongoing prior to the earthquake and prior to the cholera outbreak in Haiti. And basically we were able, with some of the first runs that we did at DTU, comparing the Nepalese cholera isolates with the ones from Haiti that were sequenced with the US CDC. And in addition to the genomic analysis, we also did PFG, malware, and traceability testing. But again, the data from genomics were basically at a very much higher resolution than PFG and all of the other analysis that we did. So that was sort of the first evidence that we had, that just with a simple SNP analysis we were able to create a phylogenetic tree with the Tahitian isolates placed within a Nepalese clade. So we were able to see and identify 1 to 2 SNPs difference between Nepalese and Tahitian genomes. So that was really the first evidence that we had that genomics had this immense power in surveillance and outbreak investigation. So moving up till today, we of course have a general interest in helping low resource countries. So this is part of our mandate as both a WHO collaborating center, FAO reference center, but also EU reference centers for AMR. So we are very keen on establishing whole genome sequence based surveillance in low and middle income countries. So we were lucky enough to be tasked by the UK aid, the Fleming Fund, to establish sequence centers in Africa. So basically, the aim was to generate data for action on the continent, so meaning generated in Africa and for the continent. So basically allow the continent, Africa CDC and the partners to translate the data into action. So the project scope was basically to build capacity for whole genome sequencing, allowing African scientists and risk managers and assessors to conduct whole genome sequencing on primarily bacterial pathogens for AMR. And the goal was to establish 3 regional centers, have genomic surveillance for AMR in place, and sort of establish a referral system and sequence at least 15,000 genomes and do a lot of in-person training. So I'll have a few slides showing some of the accomplishments. So first of all, our starting point in Africa was basically very, very limited genomic surveillance capacity. There was a few countries that had a sequence platform established, and most of it was just for research, and also SARS-CoV-2. So SEC-Africa started around 2020 with the start of the pandemic. So there was also very minimum infrastructure for whole genome sequencing, and there was a few trained bioinformaticians in the continent. And procurement of reagents and platforms were just a nightmare. So it was very, very weak in terms of supply chains. So basically, after 5 years, we have now, when I say we, it's not me, it's not Denmark, it's actually our partners in Africa. They have generated 36,000 genomes originating from 25 countries in Africa. I think that's fantastic. I'm really proud of being part of this. So out of the 36,000 genomes, about 10,000 were SARS-CoV-2. So you probably all remember that a lot of the data with variants came from South Africa, and SEC-Africa was funding 1/3 of all of that data that came out of South Africa that we all used for identifying variants and trying to mitigate that in our home countries. So again, this had really shown the effect of sequencing. But we also sequenced more than 130 different bacterial species. So quite a broad area of genomics. So what is also important is, of course, to share the data, just as I mentioned with the philosophy of the genomic, the global microbial identifier. So we have managed to submit and share 80% of all that data that was generated in global repositories, which is now accessible for the global scientific community. A lot of the data, unfortunately, have been human clinical isolates. So as you can see, we only have 8% that are animal and food related, and only 4% that are environmental source. So going back to Sofia's presentation with Denmark, that the Achilles heel in the Danish monitoring were the environmental part. But basically, we are not alone in Denmark. This is globally. So basically, a lot of the resources goes to the human sector, and that also means genomic funding goes to the human sector. And it's maybe also easier to get strains from the human sector because it derives from all the hospitals, where the animal isolates is much scarce, and they are not collected in the same way. And environmental strains are just absent. That's at least my experience. And this is, of course, not an excuse, but basically the area that we need to focus on in the next sort of years to come, as also Sofia mentioned. So when you set up a genomic surveillance, you also need to make sure that you trust the data. So as we have done, and I showed with the phenotypic data, you also need a proficiency test or external quality assurance scheme on a genomic level. So we have built this system also at DTU that we have offered the EU labs and also the low and middle income countries in Africa and in Asia, basically where we send out a live culture that they need to sequence. And so that includes, of course, DNA purification, library prep, etc. And then they would sequence it and submit the data, and we would assess the quality of that data. But it also have a dry part, which basically is that the user also need to type their own data. So they need to detect resistant genes, but also predict the phenotypes. So basically saying, if they have these resistant genes, what would they confer resistant towards, and build sort of a phenotypic profile. But there would also be other typing parts, like detecting plasmids, MLST types, etc. So this is sort of a second component to the proficiency test. I have marked this in because this is also going to be offered through the PulseNet system, through the US CDC, that have less funding now after the new US administration. So we are trying to collaborate across, so they are likely going to adapt and integrate this last dry part in their sort of PulseNet assessment. But as you can see, in 25, we have provided species for MRSA and Klebsiella and also Campylobacter. Previously, also E. coli, Enterococci, and Staph aureus. But as I said, we also covered sequencing for a lot of different countries in Africa within SEC Africa. So this was building the referral system. So basically we had not only 3 sites, but actually at some point we had 5 regional centers operating. And we had basically an offer that any scientist in Africa could submit isolates for sequencing. So we would cover the expenses of the whole genome sequencing if the provider would cover the shipment and the bacterial diagnostics. So this was a great offer, and it was heavily used. So we also showed that it was actually possible to build sort of a referral system. We also wanted to sort of democratize sequencing, so not only with regional centers, and that was why we saw the benefit of also deploying MinION ONT sequencing for frontline diagnostics. So basically we provided both protocols, but also a setup for central site sequencing. So we piloted that in South Africa with one private and one public clinical laboratory, and then basically have them in real time sequencing all their salmonella isolates, and then at the same time have it sequenced. At the reference center in South Africa to compare the quality and the data, and it was quite successful. So we also repeated that in Ghana and Senegal in a similar way, and this just show that that you can actually both have a central regional site that do a lot of the more sophisticated analysis and bulk sequencing, but you could also deploy sort of central site sequencing with a less throughput setup. So this is where a MinION is ideal with a laptop sort of unit. So basically what you need is a MinION and a laptop, and then you would be able to run it. So of course, all of this needs personnel and staff. So we did a lot of training and workshops. So we trained during the 5 years almost 200 individuals when we did training in Southeast Asia. And as you can see in the illustration, this is just some pictures. The middle one is from ONT, central site sequencing, looking at the MinION run. The first one is about assessing the library quality, and then the last one is again, as you probably also know, a spanning tree looking into the interpretation. So what is also important is, of course, in a low setting context, how do you save money? Because again, sequencing costs money. So we actually set up a webinar series where we focused on which isolates do you sequence? How do you select the isolates that you want to sequence? What metadata would you need, and how do you communicate your results to those that need to take action? So we have all of these webinars available where we have invited experts to give their pitch on these issues, because this is not trivial, and genomics is expensive. So it's really important that you have clarity about which one would infer data that can be used for action, and which data would not add anything more to your genomics. So that's a way of saving funding as well. We are of course going to publish this data as well as an expert elicitation. We are also continuing with setting up simulated exercises, because again, when you have an outbreak, which is fine, but in peacetime, you also need to make sure that you know who to talk with. Just as Sofia said, in Denmark, we have the weekly meeting within the outbreak group, and this is something that we also need to establish worldwide. So basically setting this up, not only in Denmark, but in other places. And this is where it's important to run simulated exercises. So this would be establishing teams of bioinformaticians, microbiologists, and epidemiologists, and then basically solve an outbreak in real time. This is what we have done, and we are still continuing to offer these exercises. So when you also need funding, then it's very important that you also communicate to your upper management level what genomics is, because very often it's your general director that would give you the money. And if that person do not know about the added value of genomics and the benefits and understand the concept, this person might not be funding your activity. So we actually developed a small course. I think it's a 3 hours course for basically upper level management to basically make them understand the basic of genomics, but also the added value and benefits of it and the cost saving. So this is just an open invitation to log on to this free of charge open access course. that would allow you to understand genomics in a very basic way. So when we talk about SEC Africa, there was, of course, also some factors that facilitated the change that we saw in the African continent. So first of all, we had some strategic funding and partnership in place. So there was a lot of funding coming from the UK Aid Fleming Fund that was funded by the Ministry of Health. And that, of course, opened some doors with collaboration with Africa CDC, WHO, FAO, and a lot of other major stakeholders in Africa. But we also opened up for this regional hub model where we established regional centers rather than try to set up sequencing in each of the lab around Africa. And then we wanted to also expand on the infrastructure. So basically do bulk procurement of both the sequencer but also the ancillary equipment rather than doing a minimum purchase. So basically do bulk across our partners. And then, of course, also be flexible. So again, we immediately directed some of the resources to the SARS-CoV-2 sequencing during the pandemic rather than just continue with AMR. And that's also a very crude selling point when you want to establish a genomic surveillance system. That again, this is a method for all, so it's universal, and you can easily change from one pathogen to another. So this is really important when you that you have this flexibility and adaptation in place. And then, of course, have a community where you discuss some of the challenges, but also some of the gains. So setting up a community of practice, sharing what you have learned, build local champions, early career scientists, etc., is really important, because if you keep the information to yourself, the initiative would basically die out. And then again, make sure that you use the data for action. So basically contribute to investigation, stewardships, surveillance, and of course also make sure that your data are published. But I also have identified a ton of barriers for the genomic implementations. So I just have these slides that summarize everything. First of all, we need country buy-in and funding. So again, you might have funding from a grant, but that will always end. So you need sustainable funding, and it has to be from the local government. If they're not funding it, it's not going to be sustainable. WHO, FAO, or Gates, or other philanthropic donors don't have enough money for this. It needs to be funded nationally. You need, of course, to also acknowledge there would be competing national priorities. We talk about AMR, but it actually might be that HIV or malaria is a much greater problem in your country, and we have to accept that. We have to work with the supply chains. So this is really important, and they can be influenced. If you are more than yourself and you form a network, you can actually influence it. When we started SEC Africa, there was no Illuminate representation in Africa. After 5 years, there was one central supplier, and then they had 10 channel partners scattered around Africa. And that was because that there was a network that boosted and pushed for this procurement and supply chains establishment. As I mentioned, a lot of the data were clinical simply because we were lacking isolates. So you cannot expect to set up a genomic surveillance if you do not already have a pre-existing phenotypic surveillance. You don't basically have the right isolates. So you need a pre-existing AMR surveillance to build upon. And that was also the reason why we didn't have enough of the animal and environmental isolates. Then you need to build trust among collaborators, and you need to share data, because if you keep your data to yourself, you wouldn't be able to really get the full benefit of it. Then you need to address staff retention. You need to keep educating, train, and do knowledge transfer. And then you, of course, need to invest in IT infrastructure. So it's not cheap to do sequencing, so there have already been a lot of comparisons about cost. There's actually in the second column, this is Argentina, that basically cost estimated what one sequence would cost based on Illumina, and that was about these 150 euro, which is far away from the 50 euro that we forecasted in 2011. But I also think sequencing is expensive. We have calculated our cost, and it would be approximately 50 euro right now to do in-house sequencing. But we are also outsourcing our sequencing. If we have bulk isolates, I don't want to spend money and time on this. I want to basically outsource this. And there's a lot of different vendors that can sequence for you. This is one example that we use. This is GenoHub, which is a US sequence broker, meaning that you send them a quote and then they would find the cheapest solution. And I just have a summary of our last quote. We wanted to have 600 isolates sequenced. It would be sequenced on a Illumina NovaSeq 6000. We would have sufficient data in reads. We would have about 1 gig of data, and look at the cost, 15 US per isolate. And if we want them to do library prep, it would be another 4 US. So about 20 US for one genome and a turnaround time of 30 days. I can easily wait for surveillance data for 30 days. It was a bit different if this was an outbreak, but for surveillance, why not wait 30 days? And in my mind, or with our cost, save 30 US per genome. So this is definitely something to consider as well. Sofia already mentioned a lot about the EU surveillance system, so I'm not going into a lot of details here, but basically it's a One Health system, and Denmark is based on basically the mandatory part of the EU surveillance. The EU surveillance is based, especially the food and the veterinary part, on an implementing decision. So every 7 years there would be a new legislation. There would be a new one coming up in 2028, and already now, even today, I had discussion with EFSA about how to modify the current surveillance system. It always takes a couple of years to plan the new legislation. But basically, as you can see, our EU surveillance consists of both data from humans and animals, just as Sofia mentioned, and it all goes into the summary report. And I have another slide that also Sofia showed, so I'm not going into details, but I'm just going to say that what we sequenced in EU is only the ESBLs and AMC and carbapenems. All of the rest is basically still phenotypic susceptibility testing. And as I said, we are now discussing the next part of it, and I would doubt that everything would be transitioned directly to a complete whole genome sequencing, because among the EU countries, only 10 of those, approximately 10, would do sequencing on a regular basis. The other 20 or more, they don't have the capacity yet, because sequencing in Europe have been in academia for 15 years, but they have only recently emerged in public health and veterinary health. So again, EU is not ready for full-scale genomic surveillance yet. We have done a few baselines. There have been, last year, we had a baseline for MRSA, where we surveyed MRSA among pigs. And here, as you can see in the last bullet, we screened all the isolates with PCR, and 20% of all the strains that were positive for MRSA were also sequenced. But the problem is that when you do sequencing, you get a ton of data, as I mentioned. And this is just part of the mandatory genomic characteristics that the member states in EU had to report. And that was just way too much. So basically, this is the comprehensive scheme that EFSA offered. And this is not trivial to fill in. So basically, what EFSA have done for the genomic surveillance is not to collect the genomes themselves, but basically have all the member states to convert the bioinformatic analysis. So basically they have identified this gene, and then they would convert that gene into a code. You can see it's called a SSD code, and then report these codes. This means that one strain would easily end up with 20 or 25 different codes. So this is not a comprehensive and ideal way of basically reporting genomic data. So EFSA also have to rethink this if we're going to go full-scale genomic surveillance in EU. And definitely something you also need to think about starting to implement genomic surveillance in your region or in your country, so you don't end up doing the same mistakes as we have done in Europe. So as I mentioned, that in the EU we are way behind academia. So basically what we have done in Africa, training African public health and veterinary health institutions, we are definitely on the same page as we are in EU. So just recently, the public health laboratories in EU were offered a huge grant from the HERA. It's a EU grant. And then I was leading another project called the European RefLab CAP to basically educate the reference centers to adopt to genomic data. And right now we are working on the second project. It's a ECDC project called GenEpi BioTrain. Here we were commissioned to train 2,000 health professionals. So that's bioinformaticians, microbiologists, and epidemiologists in actually being able to analyze genomic data for surveillance and outbreak investigation. And as you can see, yeah, this is maybe not the right slide, but again, this was given to basically on the backside of the COVID pandemic to really set up the preparedness for the next pandemic in public health. So basically building this interdisciplinary collaboration between bioinformaticians, microbiologists, and epidemiologists. And we're not only focusing on AMR. So basically it was set up as semesters for the 4 years. This is a bit of an old slide, so the to be decided have already been done. But basically we set it up with this we call pathogen waves. So the first one was, of course, training SARS-CoV-2 and influenza. Then we moved into another pathogen wave of AMR, and then foodborne and waterborne diseases, vaccine preventable diseases, tuberculosis, etc., etc. And the training consisted of different workshops. So we started basically with bridging the gap, educating bioinformaticians. Then we set up interdisciplinary training where we basically built country teams of bioinformaticians, microbiologists, and epidemiologists, and then we start training them on outbreak investigation and surveillance. And I won't go into details here, I'm a bit behind with my slides, but basically here you also see sort of the training concept. So some of the training components were 2 weeks, there was exchange visits, there were some parts that were for teams, the country teams, others were more for advanced bioinformaticians, and again others were for novel and new beginners. All of the training material that was provided in the GenEpi BioTrain have been deposited in the ECDC EVA Virtual Training Academy. So this is free of charge. You can log on and you can see all of the data. There's actually a virtual training on the cholera part that I just explained earlier on. So there was actually a bioinformatic course on that as well. But this EVA platform has been visited by 14,000 health professionals already, doing a lot of the training by themselves. So this is definitely a resource that are worth to visit. So coming to an end, I just also want to announce another project. So this is funded by the Novo Nordisk Foundation. This is a 26 million euro investment to build up a genomic pathogen analysis platform, or just GPAP. So basically, this is in recognition that a lot of the gaps in sequencing for surveillance is not to generate the sequence itself, but it's basically to understand the data. So basically, the motivation is the same as I started talking about, that we have this global serious threat of infectious disease, and the surveillance is not timely, and we need to take action. And the global surveillance system as it is today, especially in low and middle income countries, are weak and fragmented. So basically, we have an urgent need to establish a One Health genomic pathogen surveillance, and this is what GPAT is all about. But it's also to make sure that we strengthen and build a robust system. And the methodology is basically to set up a platform for online bioinformatics and epidemiology analysis. So basically develop a secure space where people can share the data, they can store it. So basically we don't have access to it at all. So it would be the submitter that entirely own the data and only have access to that, unless they share it with colleagues and build small sharing groups, but they can store it free of charge and they can use the analytic part. And this is basically what we are working on for the next 6 years. And the beauty is that you just submit your data and then the platform would start analyzing. You don't need to choose the tools yourself. They would basically assess the tools with an AI component that basically would analyze your data without you telling what it is, and then start analyzing. And then there would be a chatbot where you can start asking questions. What does it mean? How do I take action? How do I understand it? What do I need to do more in terms of analysis? So this is also agnostic, taking bacteria, fungi, virus, metagenomes, and then it would convert everything into this bioinformatic analysis, and then basically have the AI to steer and manage everything in terms of visualization, browsing, etc., and make sense of the data. But it also have, of course, this very important layer of sharing and storing. And we're not building this ourselves. It would be a co-creation with low and middle income countries. So I already mentioned that this is a 6 years roadmap. I'll move into this. What is also important is that we are not doing this alone. We would recruit up to 15 partners in low and middle income countries. We have already identified SE-COMISCA in Mexico to really help co-develop and co-create this platform. Coming to a very end, basically genomic have shown its advancement, and we are already starting implementing genomic-based AMR surveillance, which would be essential for future prevention and control strategies to really engage with the threat of AMR. And we can see the large added value of genomics, especially with the added value in terms of understanding the emergence of AMR, translating it into action. And of course, we need to basically coordinate and collaborate across with stakeholders to make sure that this is not built in silo, and we need to build the capacity in global national reference laboratories to the same level as we have seen in the last decade in academia. And we also need, of course, to establish the phenotypic surveillance system, because without that we would not have a genomic surveillance. So we need to allocate accurate resources. We need to build the infrastructure, provide the protocols, data sharing mechanisms, and so on. But we also need, as I mentioned during the talk about barriers, we need the political investment and the buy-in, because if we don't have that, we would never be ready for the next pandemic. So right now we have the large dependency on temporary research grant, and we need to go beyond that. I'll stop here. Sorry for going a bit over time. Thank you.
Muchísimas gracias, René, por la excelente presentación. realmente mucho aprendizaje y vamos a pasar ahora a la parte de preguntas. No sé si Tabata o Esmeralda vieron alguna pregunta en el chat. Si no, podemos preguntar a Eileen si se mantiene la pregunta que ella tenía, había levantado la mano.
Hola, hola, buenos días, muchas gracias, Antonieta. Sí, de hecho veo que muchas gracias a Sonia porque ella me la respondió también en el chat. Yo había preguntado justamente que si para la vigilancia de productos de importación, cuáles son las acciones tomadas en caso de obtener resultados de preocupación de genes circulantes de resistencia o algún tipo de resultado de preocupación. Bueno, yo trabajo en el Servicio Nacional de Salud Animal y aunque si bien es cierto, nuestra vigilancia radica principalmente sobre la salud, la protección sobre la salud pública, también tenemos también una responsabilidad sobre la salud animal siendo un concepto de salud pública veterinaria. Sin embargo, nosotros también manejamos mucho el concepto del comercio. Y es por eso que justamente le preguntaba a Sofía esta segunda etapa de la pregunta, porque ella me responde que efectivamente ellos cuando detectan algún tipo de situación preocupante, ¿verdad? Se comunican con la administración veterinaria y alimentos danesa especialmente si está relacionada con productos importados y toman las medidas como parte de sus autoridades, ya sea, por ejemplo, investigando al productor o contactando a las autoridades de los países. Pero entonces le preguntaba a ella adicionalmente, y aquí viene la segunda pregunta, en concepto, si se ha llegado a tomar en algún momento alguna medida punitiva o sancionatoria, dependiendo del riesgo que esto represente, por este enfoque comercial que nosotros en términos del comercio que tenemos justamente de productos cárnicos y otros productos, inclusive vegetales en un futuro, ¿verdad? Solo que hoy no lo vemos aquí, se podría dar, ¿verdad? Esto porque siempre ha sido un tema de discusión y hay un temor en el aire justamente en nuestro sector de no publicar datos por ese temor, ¿verdad? A algún tipo de medida punitiva o algún tipo de acción que pueda darse. Nosotros tenemos claro que la vigilancia es un proceso constructivo, sin embargo, por eso es que nos gusta como sondear, como para aclarar esa posición. Y bueno, ya le cedo el espacio a Sofía. También tenía una preguntita o comentario para René, pero no sé si habrá tiempo para hacerlo. Gracias.
Sophie.
Thank you. Yes, thank you for your question, Aileen. Sorry, I didn't see the follow-up in the chat until now. So I think I can give you an example of actually a real situation that occurred where we detected a few years back a high occurrence of ESBL E. coli in a category of imported meat. And when I received the data before reporting in Denmark in more or less a raw state, so I could see that most of the isolates contributing to this increase were coming from a given country. Now, my role working in Denmark and being part of academia and advisory is not to do risk management, is to do risk assessment. So it's part of that role to inform the authorities who are the risk managers. And in Denmark, there's this clear distinction between risk management and risk assessment, as it should be. So I'm not to say what the action will be, but I'm the one to tell the risk managers in case there's evidence, any concerning evidence, which was the case. So we did have a meeting to discuss this particular result, and the authorities were informed and alarmed, and then they took action. And I totally agree that, of course, surveillance is not detached from the rest of the reality. So we need to, of course, account for the fact that there's trade involved. Obviously, there can be diplomatic issues. So we discussed to what extent the information should be given to the public in Denmark. And then fortunately, I think we were in the position where we usually don't disclose the origin of the imported samples. We just classify it as either domestic or imported meat. So we provided, so to say, the truth, but not the full truth. So we did not disclose the particular country that the samples were coming from, because by the time the report was published, the authorities were still handling the situation, contacting the producer in the foreign authorities to see what was going on. This was also in the meat category, which was turkey meat, which was had just become mandatory to monitor for this type of resistant bacteria at EU level. So it was new data. It was not data that was there previously. So we didn't not have a baseline to compare it with. So this concerning finding was actually not only in Denmark, but found in many EU member states. So we were all on the same page, and I think all the countries were trying to address the issue at the same point, at the same time. So again, coordination at EU level in that case was very important. And now, 2 years later, when we had data from imported turkey meat again, we could see that something had happened because the origin of the meat was the same and the levels had been brought down of occurrence of ESBL E. coli. So what we provided to the public in the Denmark report was that we found this concerning finding that there's a high occurrence of ESBL E. coli in this particular meat. It's imported, and X% of the samples were from the same country, but we did not disclose which country it was. So we found some sort of compromise to keep because then we could be stepping into a management role by disclosing that type of information. So we kind of protected what was happening at management level in that way. I don't know if this answers your question, but we did have the dialogue with the authorities to try to find a compromise in a way in which we still inform the public and disclose the findings in the most transparent way, but without compromising trade or diplomatic relations with the country that had exported the meat to Denmark.
Gracias. Vamos a ver si habían más preguntas, me parece. Tabata.
Perdón, sí, tenemos una pregunta en el chat, acaba de entrar, está en inglés, no sé si la leo en inglés.
Sí. Dale.
Entonces, la pregunta viene de Amaris Contreras, dice, depending on the health situation of the country of origin and the country of export, surveillance will be carried out for any pathogens of interest. Should I repeat it? It's in the chat.
And
I
who is
guess,
the...
yeah,
I don't know if I can repeat it, or I don't know if René want to address it or you.
Yeah.
It's almost at the end. I will repeat it. Depending on the health situation in the country of origin and the country of export, surveillance will be carried out on the pathogens of interest.
So
this could be related to what you presented, René, I think.
Yeah. Again, so depending on the health situation, if you see an emergence of a certain pathogen, you need to establish the surveillance of that. I don't know if that's sort of the question, but just as we had the MRSA baseline in Europe, that was to get a bit more understanding of the circulating clones from MRSA. We would now also start looking into aquaculture, so basically assess the occurrence of Eomonas and Vibrios in fish and shellfish. And that is also to see if there's anything we need to be concerned about in terms of a health situation. So it might be that this is the decision of establishing a surveillance system is based on known issues or concerns, and then you need to set it up. But it could also be that in relation to the international health regulations, that one country would be, just as Sofia said, would be affected by a commodity or infectious disease posed by another country. And then you might direct that question to the other country and basically ask that country to establish the surveillance or at least help in understanding the emergence of that pathogen in the other country. So I think that's probably my best answer if I understood the question right. But basically also in Europe we have, as also Sofia mentioned, we have this emergence of carbapenem resistant among food producing animals. And as you know, carbapemins are not allowed for animal use. So we are trying to establish a new surveillance really to figure out where does this come from. It likely is a human to animal transmission, but what are the intermediate steps? What are the vehicles for the plasmid transmission from humans to animals? So again, and this is also involving other countries because it might be that we import commodities that actually have carbapenem resistant Enterobacteriaceae in our food, and we want to avoid that. So this is some new activities that EFSA have initiated, both in Campylobacter but also in Enterobacteriaceae. I think I'll end here.
Por acá tenemos, Tabato.
Iba a
Gracias.
decir que tenemos más preguntas, no sé cuánto chance tenemos para preguntas. Ya se están volviendo no tan tímidos.
Sí. Si
Ajá.
quieres, vamos a ir preguntando algunas. Por acá veo una para la doctora Ribeiro. Desde su experiencia, ¿cuál considera que es el factor más importante para avanzar hacia la integración de la vigilancia y una salud en un país? Y si tiene alguna experiencia o ambos también hacia más la parte de, veo que África, porque han trabajado o Latinoamérica, se los agradezco. Gracias.
Sorry, I'm a bit lost reading all the questions. I think the question was what I consider most important to integrate or implement integrated surveillance.
Yes.
Yes, I think, of course, the resources have to be there, and I can't go back to you, so I can see you now, but of course the resources have to be there. There has to be a governance, and again, as I mentioned in the presentation, I think one of the big challenges to bring the environments into the Denmark is that there is no governance. There is no, I mean, the funding, the authority that will be standing as data owner and provider for such a program, such a part of the program is not there. So there has to be a governance in order to resources to be there for people to actually be hired to do the work. And then, so that the governance is important to fund such a program. The next factor, I think, and a determinant one, is the operational level, in the sense that if the sectors work independently and in a silo approach where they each do their own thing and and then put everything under one report. I don't think that's integrated surveillance. So they really need to work as a team and have the time to sit regularly together and really produce one report, or it doesn't need to be a report, but that they work as a team, despite the fact that they work with different data sources and with different sectors, but that they put a common effort into doing something that is as integrated as possible. Of course, there will be parts where the data is not comparable, the pathogens are different, the methods are too different, and things cannot be compared. There will always be parts that are sector specific, but if the teams do not sit together and work harmoniously and to a common goal, then integrated surveillance is not possible.
Ujú. Gracias. Yo creo que uno de los aspectos muy importantes y verdad que se va repitiendo y se dan las dos presentaciones es no solo esa parte técnica y el trabajo entre humanos, sino ese apoyo político que se necesita. Veo algunas preguntas por acá también que van direccionadas a eso. Ahora, ¿cómo se logró la regulación de lo que fue el plan de acción de Dinamarca? Y quería aprovechar la pregunta que veo por acá, que tiene que ver mucho con la parte de gobierno, pero también los comentarios, porque creo que también y todos los que estamos conectados tenemos alguna función en resistencia antimicrobianos y en la vigilancia. Y al ver la presentación y donde se menciona la herramienta para evaluar el sistema y ver si realmente es integrado el EPICAP, sorry, em, quisiera saber su opinión sobre, a pesar de no tener un sistema integrado que aún no se puede evaluar, ¿qué piensa de usar esta herramienta para utilizarla de apoyo para identificar qué se tiene y qué falta aún por desarrollar en un país? Y eso va a incluir políticas o la parte técnica, está incluido en esta herramienta. Y algo más que tiene que ver con gobernanza, me parece a mí, es que desde la parte técnica, lo que es las autoridades de salud nos preguntan, bueno, y para implementar o de dónde partimos, qué indicadores utilizar. Y eso lo preguntan en las diferentes áreas. Para dar un ejemplo, sabemos que desde la Organización Mundial de la Salud y la ONU se habla de los indicadores de magnitud como es staph meticilino resistente, como es resistencia a cefalosporinas de E. coli. En este aspecto quería preguntarles si ustedes como país en Dinamarca están utilizando estos indicadores a nivel de país, si no cuáles, y sé que estos van direccionados a lo humano, y si tienen algún tipo de indicadores que se pueda comparar en la parte animal, que va a ser también un insumo importante para las autoridades y que no los piden a nosotros, al menos a los que estamos en la parte de laboratorios nacionales de referencia. La pregunta puede ser para los dos o
Yeah.
para Sofía. Ustedes son los que saben quién puede responder un poco más. Gracias.
Sofia, will you answer this?
Yeah, I can. It was a long question, so I'm not sure I really got it all, but um, yeah. So I think the indicators, or I would rather call them targets for monitoring and for following the situation in both sectors have been defined in the national action plans. Some of them are not concrete and measurable, so they are more, I don't know. For example, in the action plan for the animal sector, for the veterinary sector, there's something like increasing the collaboration at international level, which is a lot of what René is doing. So that's contributing to a lot to fulfilling that particular target in the national action plan. But then there are some measurable ones like an overall, I don't remember now, I think it's 8% over a few years reduction in the overall use in the pigs, in the pig production sector. I think we are, so that's in terms of national targets. Then in terms of indicators as such, we are very much aligned with what is done at EU level. So we monitor resistance in indicator E. coli, we monitor the levels of complete susceptibility, the levels of different combined resistances that are relevant for human treatment in case they occur in indicator E. coli or zoonotic pathogens. So there the focus is on critically important antimicrobial resistance for the human medicine. On the human sector, the latest version, which came out at the end of last year, of the National Action Plan for the Human Sector, defined actually quite many measurable targets, objectives to be monitored, and our colleagues at SSI were specifically instructed that from now on, the authorities want a section in Denmark that solely focuses on following all of those targets. So yes, answering your question, don't ask me what the targets are because I haven't looked at the list yet, and this is quite recent. It's from end of last year. But yes, there are some national monitoring targets that are part of the National Action Plan that the colleagues on the human side are supposed to monitor on a ongoing basis every year.
Thank you. Y, perdón, no sé si capté, pero de la EpiCap, recomiendo usarla como para revisar qué se ha implementado y qué se puede implementar o...
Now the WebICAP, the One Health EpiCap, the procedure was to the investigators set all the participants in Denmark and people with the knowledge in the program around the table, and they had there was a structured questionnaire that you get from the tool that all had to answer. Of course, if you already have an integrated system, then it will be more of a discussion, but I would say it's a good idea to use either this or any other tool, and there are many available, as a starting, if not for anything else, just to sit people around the same table and learn about each other's sectors and identify common points where they could collaborate.
Thank you. Estamos viendo ya. Muchísimas gracias. Bueno, por el tiempo, tenía más preguntas y específicamente será en otro momento a René porque muy interesada en la parte de fenotipo y genotipo y etcétera, pero bueno, las dejaremos tal vez para otro momento por el tiempo. Pero Tabata, no sé si identificas alguna otra pregunta para dar oportunidad más bien a los participantes.
Sí, tenemos un par más. Hay una específica para René, que si conoce de algún tipo de financiamiento para países de Centroamérica, dado que se habló como de algunas especies de proyectos que han surgido en otras áreas del mundo, que si conoce algo específico para nuestra región.
The answer is no, but you're not alone. The problem is that 1/3 of all global funding for health have disappeared. So as you know, the US was the first one to start, then the UK aid, and then it was basically all of the European countries. They have also limited a lot of the spending in public health, basically to transfer that to defense budgets, unfortunately. So this is very unfortunately, but that's how it is.
Okay, and there is as well another one that I'm gonna tell it I'm gonna just read it, and the question is, how was the legal framework that give rise to the regulation of the AMR surveillance plans in Denmark constructed? It's in the chat. I'm gonna repeat it. How was the legal framework that give rise to the regulation of the AMR surveillance plans in Denmark constructed?
Yes, here I would actually ask René whether he can answer because he's been in the game for way longer, and I think he has assisted to the start.
So Yeah, so I was also part of the DanMap group originally, and it was basically a decision that was taken by the government. So basically, there was a lot of consumer awareness, and the consumers actually had a lot of power. So they didn't want to buy, for instance, commodities like meat or stuff like that. If they were contaminated with Salmonella and Campylobacter. And that really started a lot of the awareness, and there was really a demand on setting up a surveillance. And then after that, there was the entire discussion about growth promoters for AMR, and basically those two happened these almost 3 decades ago, that the consumers that were really aware that they didn't want to have contaminated or incriminated food, and they wanted high quality food products. And that pressure basically set the government to establish the surveillance system. And then, of course, with the surveillance system, you actually also gain a lot of information. And you could see a lot, maybe Sofia didn't show it, but we have actually targeted Salmonella over the last 30 years with intervention that have really reduced it. So today we are what is called a Salmonella-free country. We have a very few Salmonella cases, but we are a Salmonella-free country. And that is something we have worked on for 30 years. So both the industry have the benefit, the public have the benefit, and the ministries, the government have the benefit, because it all ends down to sick days. So either hospitalizations or sick days. So it's a positive investment in surveillance, and that was the legal framework around it. And even the industry works as a stakeholder, that they all actually also voluntarily banned certain drugs or help with interventions. So we are all in the same boat.
Thank you. That's really quite interesting, the awareness. Oh, so lastly, I'm going to read one more. Esta está en español, estaba un poco antes. Vamos a ver si no la perdí. Esta pregunta era de República Dominicana y es como muy específica. Era que si alguno de ustedes ha trabajado con Mycobacterium bovis y tiene alguna particular, que si ha trabajado con Mycobacterium bovis y paralelamente entonces también ha trabajado con la resistencia antimicrobiana en tuberculosis humana, como relacionando como estas dos aristas en una sola salud. Como entonces el Mycobacterium bovis y la resistencia de los antimicrobianos en tuberculosis. Es muy específica.
So no, I haven't worked with TB, but I collaborate with the reference center in Germany for TB, so I can link that person up with the head of the department for multi-drug resistant TB in Europe.
Well, that was a question, a really punctuated question.
Okay.
Quite a
good.
specific one. Yeah.
Yeah.
Okay. No veo más preguntas en el chat. No sé si se me está escapando alguna. Entonces no sé si querías hacer alguna más en específica o no sé si Esmeralda quiere algo más en específico. ¿O podemos ir cerrando?
No, me parece que vamos cerrando. Más bien devuelvo a Esmeralda y con qué seguimos ahora para ir ya cerrando.
Sí, muchísimas gracias. Antes que nada, agradecer la excelente participación del doctor René, las presentaciones de Sofía. Sabemos que es un reto muy importante para los países de Centroamérica, porque muy bien lo han mencionado, a veces si la resistencia se vigila en los diferentes sectores, pero a veces se hace por separado, y lo importante que es considerarlo, la vigilancia con un enfoque de una salud e integrar toda esa información. Eso nos ayudará pues a enfrentarnos a este principal reto que tenemos ahora todos los países. Pues por mi parte nada más agradecerles y sabemos que hay más preguntas en el chat, se pusieron muy activos al final, pero en honor al tiempo y porque sabemos que en Dinamarca también la diferencia de horarios ya es tardecito, nada más por nuestra parte agradecerles en nombre de la región. Gracias al equipo de Costa Rica y a todo y a cada uno de los participantes. Antes que nada, y para finalizar, nos gustaría que los que tienen disponibilidad de abrir su cámara lo puedan hacer y podamos conservar algunas fotografías del evento. ¿Pueden abrir sus cámaras?
Muchas gracias a todos. Vamos entonces a prepararnos para tomar las primeras fotografías. Ya que somos varios con las cámaras abiertas, vamos a tomar varias y vamos a dar el espacio para que todos también puedan acomodarse. Ok, vamos entonces en este momento con la primera fotografía. Permanezcan todos, vamos en 3, 2, 1. Ok, vamos a tomar otra fotografía para que todos puedan salir. Permanezcan con su sonrisa. Vamos nuevamente en 3, 2, 1. Ok, vamos de nuevo con otra fotografía. Vamos en 3, 2, 1. Acaba alguien de habilitar más su cámara. Vamos ya casi con la última para que todos podamos salir. Vamos de nuevo. 3, 2 y 1. Ok, perfecto, ahora sí, ya hemos tomado la fotografía.
Muchísimas gracias. Gracias
Thank
por
you.
todo.
Bye.
Perfecto, muchas gracias a todos. Que pasen una feliz semana. Estaríamos compartiendo y enviándole más invitaciones para que puedan unirse con nosotros.
Gracias. Hasta pronto.
Muchísimas gracias a todos.